Good evening, everyone, and welcome to Data Matters seminar. Today, I'm very excited to introduce our speaker, JINGYI Jessica Lee, who is an associate professor in the Department of Statistics at UCLA with appointments in the Department of Human Genetics and Department of Biomathematics. At UCLA, Jessica leads a research group called Junction of Statistics and Biology, where she and her students focus on developing robust statistical and computational methods answering important questions in genomics and health. While Jessica is at UCLA, she's actually visiting us from nearby, Boston, where she is a Harvard Radcliffe Institute Fellow. In her year-long fellowship at Harvard, Jessica plans to write a book to clarify common confusions in genomics data analysis by connecting cutting-edge genomics research questions with fundamental statistical and machine learning methods. Jessica has received multiple awards and grants from NIH and NSF, including an NSF Career Award. She has also won the Johnson & Johnson Women in STEM 2D Scholars Award, a Sloan Research Fellowship, and a spot on the MIT Technology Review 35 Innovators Under 35 China list. I met Jessica during the Banff International Research Station workshop this summer, and her work on tackling the issues with single-cell data analysis and modeling, I found it very interesting and impactful. So I'm very glad that she gets to share her work with us today.
Thank you so much, RITAMBHARA, for the invitation, for the nice introduction. So it's my honor and pleasure here to present our work about the single-cell data analysis. All right. Okay. Now it's on. Thank you. Yeah, no problem. So we're very enthusiastic about its multiple usages, including like benchmarking, model inference, and also generation of in silico data. So I will talk about the data structure first. So in the field, I try to unify single-cell and spatial omics data in this unified data structure. The process data is often a cell-by-feature matrix, and I'll talk about what feature means. And also, we may have some covariates of cells. I'll also detail those. So in particular, the covariates could be about some cells' heterogeneity structures, like the cells types. These can be known by knowledge, or they may be inferred by clustering, or the cells' positions in continuous trajectories. These are usually latent but inferred, and also cells' locations. These are measured in spatial omics data, but not for single-cell RNA-seq data. And regarding covariates, they may also reflect experimental designs, like batches of cells, and usually the differences of batches will be unwanted effects we want to remove, and the conditions of cells. And here, the differences among conditions are usually the biological signals. And finally, by features, I mean it could be gene expression, which is the most common type of features, molecular features we measure, like in single-cell RNA-seq data or in spatial transcriptomics data. Or they could reflect some other features like chromatin accessibility, like the open regions in our DNA measured by single-cell ATAC-seq, or the snare-seq can measure both ATAC and also RNA, and they could be protein abundance. So you can list many and many different technologies measuring different features, but the data after you do the measurements can be summarized into the cell-by-feature matrix. So the motivation of our simulator work is first computational benchmarking. So as I said, this is a very popular field, and if you check this website, just for the computational methods for single-cell RNA-seq, more than 1,000 tools have been developed. And then from users' perspective, how to choose among those methods designed for the same purpose is a challenge, right? Especially, I think most people here may be method developers. We want our methods to be used, but what if we're not the first, right? People, if they go with citations, they may pick the popular tools that are first developed. Then where do I have a chance if our methods are later? So I think benchmarking is the way to justify that newer methods may be better than earlier ones. And the second is from a statistical perspective, inference. What can we learn from the data? So for example, given that the cell covariate is something we don't care about, but we want to be conditional on, can we say how a gene is distributed and how gene-gene correlations are like? These are meaningful parameters we can estimate from data to tell stories. And finally, this is the part I'm most enthusiastic about, the ability to generate in silico data as negative control or positive control. And this idea is from wet lab biology, where these controls, especially negative control, must be set up so people can really see, whether the results I observe from the sample of my interest is something meaningful. I feel like in our computation analysis, the pipeline is getting more and more complex. Such a control is also necessary. So all these will call for a realistic simulator with interpretable parameters. So I will open my talk by giving this story about a related topic, but not single cell. I call this a teaser. It's about this popular analysis on bulk RNA-seq, tissue-level RNA-seq. And this paper is that we found by accident that popular methods do not always work if the data has some characteristics that violate the model assumptions. So this is the analysis. In bulk RNA-seq, we usually don't have a large sample size. Like back to 2014, typically when biologists compare two conditions, they may have 3 replicate RNA-seq samples per condition. So to decide whether a gene is called differentially expressed or short as DE between the two conditions, they will need to compare the genes 3 and 3 values from the two conditions and run a statistical test, right? So you could imagine this is a challenging problem because the small sample size. How can we have good power to call this gene a DE gene? These two popular methods, HR and DE-seq2, they both assume a gene follows a negative binomial distribution under each condition, and to boost the power to compensate for the small sample size, they use this idea or technique called empirical Bayes to borrow information across genes. So the genes are jointly used for estimating the dispersion parameter and its relationship to the negative binomial mean, just to improve the variance in the estimates. So they were popular because they can give you a lot of findings from the small sample size data. And we find this problem by accident because at that time, we were testing a clustering algorithm we developed on some population level RNA-seq data. By population level, a sample is from an individual. And we want to benchmark whether our clustering method is working by comparing it to random partition. And then our criteria to see, given our clusters of samples, can we find DE genes that can explain the partitioning? But surprisingly, we find that DE-seq2 and HR can find DE genes even on random partition samples. That was the story, the beginning of the story. Then we used this data set from the Cell paper to demonstrate this idea, to demonstrate this phenomenon. So in this Cell paper, we have patients in 2 groups. Before the treatment of this anti-PD-1 immunotherapy or on treatment, 51 and 58 patients. If we run DE-seq2 and HR, the red dots indicate the number of DE genes found by each method. And then we try to do random permutation of the condition labels. So we still have 51 and 58, but labels were randomly assigned. And we run the method again. So you can imagine there are many, many possibilities of permutations here. And this gray bar indicates the average number of DE genes found on permuted data. And this indicates the standard error. So what it says is that on many permuted data, each method can find more DE genes than they did on the real data. And this is something not expected, right? Why would they find so many DE genes from permuted data? Looking into the reason, we find that the issue is regarding the negative binomial model assumption. So on these data, because samples under each group are not replicates, we cannot regard individuals as experimental replicates. As a result, we will have many genes, especially those that are frequently identified as DE from permuted data. The negative binomial fitting is very poor. And also, as a comparison for the genes that are rarely identified from permuted data, the goodness of fit is quite good. So we see this sharp contrast, and this can partially explain why these genes were found from permuted data. The reason is because negative binomial doesn't work. Okay, and the consequence of this, because if we want to communicate these results to scientists, we need to say, what's the consequence? And here the consequence is that if you just trust the DE genes found by DE6 or HR and run the so-called gene ontology enrichment analysis, you can find many functional terms that are immune response related, and they are associated with the DE genes. Then you may happily conclude that, oh, yes, there are immune response differences between pre-treatment patients and on-treatment patients, and this is something reasonable, confirming my hypothesis. However, if you now see these genes are frequently identified from permuted data, you may need to step back and double think about the conclusion. That's the consequence. So the take-home story here is that we benchmark against this classical Wilcoxon rank sum test, the textbook one, and we found that it's self-consistent. The best I can say is it's self-consistent because it doesn't find DE genes from real data, nor does it find DE genes from permuted data. It's kind of conservative, but it doesn't have this contradiction. So I think the take-home message I want to say here is that when we benchmark our new methods, we shouldn't forget classical methods, right? They are still the baselines, and we should see how well do they perform. Do we really have a rationale to justify our development of new methods? I think that's the take-home message. Another take-home message is the use of negative control. In this case, we use permutation to generate negative control because no DE genes should have been found. Then that can give us some comparison. So coming back to my main story, the simulator for single-cell RNA-seq. So my former student Vivian, we developed first version as a design, and at that time the simulator was developed to help us find the trade-off as the cell number increases. Is the discovery power for DE genes really increasing? The interesting thing here is that in single cell data, because the cells are measured together, if you measure more and more cells, they will compete for the total sequencing depth. So you get shallower and shallower image of each cell. So it's not like the classical statistical setting, larger sample size give you better power. There is a trade-off. So we want to find that optimal sample size. That's the whole idea of this paper. And at that time, because we focus on the differential expressed gene analysis as before, as the one I showed before, here's in single cell, but we look at one gene at a time. So we focus on this model that focuses on marginal modeling of a gene. So the pros I can see is that it has interpretable parameters, and we can change the cell number because we consider cells as samples from a distribution. So we can just draw different sample sizes. We can allow the parameter to reflect sequencing depth. So that's all possible. And so because of that, my other former student, Miles, used this to benchmark this popular task, which is called doublet detection from single-cell RNA-seq data. And we use the simulator to generate data for benchmarking. But the cons of assay design is also obvious. First, it cannot capture gene-gene correlations. It models genes independently. And second, we did the modeling on transform data, not on the count data. We use Gaussian distribution on log transform data. These are the disadvantages we want to address. So we wanted to develop something better. And at that time, these are the simulators, maybe not a complete summary that has been in the field, but most of them didn't capture gene correlations. And our motivation was to capture gene correlations. And that came to the second version, SIdesign2. So here, what we aim to do is to fit a multivariate joint distribution of multiple genes per cell type. And from this distribution, we could do sampling to generate synthetic cells of any number user specified. And here, the sequencing depth will be reflected in the joint distribution parameter. So we could scale the mean parameter accordingly to reflect the sequencing depth. So the data could be used to guide experimental design, like the sequencing depth, number of cells, and also can be used for benchmarking. And I want to acknowledge related work that also aim to capture gene-gene correlations, but the difference is, one, they may not allow the cell number to change. If they fix the cell number, then it's a different formulation. And second, they may require additional input like gene correlation as known information, while we estimate that from real data. So I will quickly go over the method. So the notation here is for a count matrix, rows as P genes, columns as N cells. And we assume here the cells are divided into K cell types. So we have a sub-matrix just for the cell type. And our goal is to fit a parametric probabilistic model of joint gene distribution for each cell type K. So from now on, my discussion will only be for this one cell type. And please note that here, if the cells are not labeled with types, we will use clustering as the first step. But this issue will be addressed in our version 3. Okay, so in the version 2, we actually have one cell type here, and our idea is to first fit a marginal distribution for each gene I. So let's say this is a cell J. It's a p-dimensional vector because we have p genes. So here xij is the observed count of gene I in cell J, and then we want to model a marginal count distribution for gene I. So based on our empirical evidence, we only consider 4 parametric count distributions, Poisson, negative binomial, and they're in zero inflated versions because empirically we found them to fit data very well. And the key challenge here is the joint distribution. And to make sure that we can flexibly model the joint distribution by allowing genes to have different marginal distributions, we use the copula framework, which is a multivariate statistical technique. So the idea is that once we have the marginal distributions, we just transform every gene's count by applying the marginal cumulative distribution function, short as CDF. So we know that every gene after transformation will become a uniform random variable between 0 and 1. Then the copula function C will link those P uniform random variables into a joint P-dimensional CDF. That's the use of copula. So in other words, the intuition is that we separate the estimation of gene-gene correlation from the marginal distribution. Okay, and the simplest copula is called a Gaussian copula. So in other words, let's say after we transform every gene by its marginal CDF, so every variable here, the U variable, is uniform 0 to 1. What Gaussian copula does is that it applies marginally the standard Gaussian inverse CDF so that this transformed variable becomes standard Gaussian. We have P standard Gaussian. To link them together, we just need to estimate the correlation matrix. So the correlation matrix is what we need in the second step to estimate. But here, one thing we need to address technically is that Gaussian copula use this as the joint distribution. The values we sample will be continuous values, while we need count data, right? Our real data are count data. How do we address this gap? So to address this gap, we actually try to modify the marginal distribution of gene I into a continuous distribution using this technique called distributional transform. So what we did is, this is a count xij. We just take its value and its adjacent value, take their CDF values, and randomly do the interpolation by adding this random weight. And this can make the CDF function continuous. Then it makes more sense to use the Gaussian copula. So that's the idea behind it. And once we have the real data using this model, how do we fit it? We just estimate every gene's marginal CDF as Fi hat. We apply this transformation to make it continuous, as I said. And finally, this variable is uniform 0 to 1. Then we apply this inverse standard Gaussian to do another layer of transformation, and we have such a p-dimensional vector for every cell j. And pulling those vectors across cells, we estimate the sample correlation matrix. So this is the estimating step. And then when we simulate synthetic data, synthetic cells, we just take the fitted joint distribution, and the cell type proportions could be from real data or could be user specified. And then users can also specify the number of cells they want to simulate and the total sequencing depth. That will be reflected in the Fi hat parameter. And then together, we will be able to generate a synthetic gene by cell count matrix with the same K cell types as in real data, and we could also output the model parameters. Okay, so this is how the results will look like. The training data is this one used for model fitting, and this is the held out test data we didn't touch, and this is the synthetic cell. So here the number here means how well do synthetic cells resemble real cells. So it's actually a mixing index. What it actually does is that taken the two cells put together as here, for every cell we pick a neighborhood, and in the neighborhood we just count are the proportions of real cells and synthetic cells 1 to 1. If that's the case, this number will be 2. Otherwise, if the neighborhood has just one set of real cells or synthetic cells, this will be 1. So the values between 1 and 2, closer to 2 means the mixing is better. Okay, so we also did a comparison by using the model fitting without copula. Then genes are independent. So you can see now the cells, synthetic cells, are not realistic. So copula works. And these two simulators, I think they are, ZIMWave is not primarily a simulator, but a dimension reduction method. But some people use it for simulation, so we include it. And SparSim is another method. So although these two methods didn't specifically capture gene correlation, but they didn't assume independence. So we included them here for comparison. And also we can see that they sort of capture the relationship, but the difference is that they didn't consider cells as random observations. So again, they consider cells as fixed as well as genes. So I think that's probably one of the reasons, the formulation, that they are not being as realistic as we are. So I think this is the paper, right? We have published this last year, and you can check more details inside. But again, we are not satisfied here because it requires cell types to be predefined. And that's not usually the case. So motivated by this, we want to go further. And here comes to our version 3, SDN 3. And regarding the simulation similarity, now we can do more. Besides cell types, we could generate cells in continuous trajectories. We could even generate cells with spatial locations in spatial omics data. And besides RNA-seq, we could generate ATAC-seq, site-seq, methylation, DNA methylation, other feature modalities. And furthermore, we could incorporate cell covariates to reflect experimental design. Like the cells can carry with them some covariates like the individual's gender, age, something else, characteristics, or conditions. The cells come from disease condition or health condition, or even the batches. So these can be reflected in our model. So specifically, regarding the continuous trajectory functionality, we generalize our model, the marginal gene distribution model fitting, from one distribution per cell type to a regression framework. So that's a generalization. And then you can see that here the synthetic cells are more realistic because they capture the continuity in real data, while in SDN2, we have to pre-cluster cells and it's less realistic. And another functionality we added is to add this technique called Vine copula, which we actually learned because of this project. So we can output a likelihood, and we can make use of a likelihood for interpretation. I'll talk about that next. So this is the result for spatial data simulation. what we did is that we allow every gene whose mean across the space to be a smooth function. And for that, we used the technique called Gaussian process for that purpose. So you can see that actually it mimics data really well, and the smooth function itself can be subject to interpretation. And finally, my other student helped us to go beyond the counts that can be simulated, because these are based on counts. So we can get the counts for ATAC-seq data, and we can go beyond counts to generate sequence reads, the raw data. So this can be used for benchmarking some low-level tools that start with reads, not just counts. So these are simulation functionalities. For interpretation of real data, because our model is kind of parametric and easy to understand, so we could give you every gene's distribution in 2 cell types, right, for interpretation, or how a gene's mean expression change along some pseudo-time inferred from data or in the 2-dimensional space. And as I said, we could output the likelihood, and the likelihood will give us a way to do model fitting assessment. So here, the BIC stands for Bayesian Information Criterion. It's a penalized log likelihood. So using this, we can actually say if we have 2 pseudo-time inference software that infers cell pseudo-time in 2 ways, which one fits real data better? And the smaller the BIC, the better. So we can tell you this. And the cluster is the same. Given 2 sets of clustering results, we could use each of them to fit our model and see which one fits better. So I think the advantage here is that us calculate the BIC values don't require ground truth. So we don't need to know which are the real cell types. We just evaluate the model fitting. And finally, model alteration, which I think is what we need for setting up in silico negative control. Let's say this is how we fit a gene on real data into cell types. What we could do is that we could keep the results first generation, simulation, or we could manually set the gene to have the same distribution in 2 cell types. So then this is an in silico control scenario. Or this real data has 2 cell clusters, but we could pretend that there's only 1 cluster, fit our model, and then the synthetic cells from our model, null model, will only have 1 cluster. So we could generate in silico nulls in these ways. So here are examples about the model inference. So these are the genes which we fit their expression mean function along cell pseudotime. So the smooth function will capture some trends. And then this is how the Gaussian process will give us in the 2-dimensional space. The mean function is a smooth version of real data, and we could also see the trends. And these are what we could get for gene-gene correlations from Gaussian copula. So that's the Gaussian correlation matrix in each cell type. What we could see here is that the correlation matrices are similar if the cell types are similar. And it becomes more different if the cell type is more different. And the Vine copula can be interpreted as a sparse representation of the correlation matrix. So the gene-gene correlations are represented in this Vine structure. So 1 layer for the marginal strongest correlation, the 2nd layer for the strongest conditional correlation, and so on. So this Vine structure can also be subject to some interpretation. And second, and this is what the BIC looks like. As I said, the trajectory inference has multiple methods for this task, like T-scan, Monaco 3, slingshot are 3 popular methods. If we apply them to the same data, they will give us different pseudo-time values of the cells. But what we could do is that we could use each result to fit our model, calculate BIC. And among the 3, slingshot gives the best BIC. But if we look at the data visually, you can see it roughly agrees with what we see, right? The change is more smooth in the 2-dimensional representation. So it seems reasonable. And the black dots are what we did by permuting pseudotime. And we can also see this monotone decreasing trend justifying the BIC is a reasonable criterion. And finally, this is the model alteration. Let's say the real data has 2 cell types, and we have the stimulation scheme. This is the control cells. This is stimulated cells. We can see that this gene has elevated expression in both cell types after stimulation. if we keep the data, keep the parameters, and do simulation, this is what we get from synthetic data, quite similar. We could do model alteration. So we make the gene not unchanged after stimulation in both cell types. This is one scenario. Or we could just modify one cell type and keep the other. So that's another scenario we could do. And over here is the batch effect scenario. Data from 2 batches, 2 versions of 10x genomics. We see that they do not overlap that well in UMAP. If we fit our model, generate synthetic cells, we keep the batches. But we could remove the batch effect parameter and then do sampling. Then the synthetic cells will not have any batch effect. I think this is not working anymore, but it's okay. Yeah, and finally, in the bottom right, we can have this clustering relationship, right, in the real data. But if we keep the model fitting using the clusters, yes, we can mimic real data. But if we just fit one cluster, then we can remove the gap in the cell. So all the details or more results are in our paper, which has been posted to bioRxiv. And comments are very welcome. And the GitHub is also up here. So the GitHub for the software is there. So finally, I will quickly talk about why we need in silico experiments and what can we do. So this is from the Khan Academy, showing that if you want to say watering does help with the growth, you need to have a negative control without watering, right? And so in single cell, I think the challenge, a prominent challenge is the double dipping issue. So we have to do multiple analysis on the same data before we do the final statistical testing, like the differential express gene expression identification follows cell clustering or pseudotime inference. Because of time constraint, I will focus on clustering. But for the pseudotime inference problem, we have a paper about it published, and you can check it out. So basically, we consider the uncertainty of pseudotime because it's inferred from the same data. However, we didn't completely address the problem because there might be this complete null case. Like in the pseudotime case, the complete null is that there's no trajectory at all. In the clustering case is that there's no cluster at all. If we don't know this, right, we just do clustering here on this one cell type. If you use the popular SURAT pipeline, or you just use k-means, you can force the cells to be clustered based on gene expression. And if you just, given the clusters, if you just find which genes are DE, you may find genes are D, right? Because the clusters were driven by gene expression. That's the problem. So there were some existing work that realized this problem, and they try to tackle the problem in different ways. But put it in short, they either assume Gaussian distributions for genes, which may not be the case, or they assume Poisson distribution for gene, which may not be the case, and also the splitting. If you just, I think this is a very interesting idea called count splitting. Given the cell by gene matrix, we randomly split each count into 2 halves. But we had to have some preliminary results showing that it works when genes are independent. But when gene have correlations, it doesn't work. What we're showing here is the distribution of p-values under the null case. It should be uniform 0 to 1, but when there are gene correlations, it's not like uniform. And if you split genes or split cells, it won't work either. So that motivated us to do something better than the current state of the art. So our project is called Cluster DE because it's clustering followed by differential gene expression analysis. And our proposal is to use our simulator to generate the in silico null, the negative control, and use another method we developed to control the false discovery rate, FDR. So I have to acknowledge those 2 classical statistics papers for giving us the foundation and inspiration. So I just quickly talk about what is Clipper. So Clipper allows us to control the FDR without using high-resolution p-values. So specifically, we just need to calculate a contrast score for every feature, which is a statistic. And then the contrast scores, as long as they follow this assumption that if they are from the null features, their contrast score should be symmetry about 0, That's the foundation. That's the assumption. If so, and also for the features that are interesting, they have large and positive contrast scores in the blue part. And then we could use this theory from the knockoffs paper to find a cutoff on the contrast score T, such that FDR will be controlled if you call the right tail as discoveries. So it just simplifies this whole analysis because you don't worry about p-values. You just need to construct a reasonable contrast score. So in short, to do a contrast score construction, you just need a null data to serve as contrast to your real data. So I'll talk about this. I talk about this in my teaser problem, RNA-seq DEG identification. The actual data is our null target data. We use permutation to generate null data. They could serve a contrast. And in the problem here, double dipping, we could use our simulated data from the null hypothesis as the null data, actual data as the target data. So whatever pipeline you apply to target data, let's call it T function, you just apply it to the null data. And the idea is that if you believe, if the feature is not interesting, then the difference is equally likely positive or negative, then you can satisfy the theoretical assumption and apply Clipper. And the good thing about here is that you just need to generate null data once. You don't have to repetitively generate data. Otherwise, it will be computationally intensive, and we save that burden. So this is the Clipper paper. You can check the details there if you're interested in using it. And finally, to this problem, right? This is the real data. If we just use permutation of the label in this case, I'm showing that it doesn't work because the cells still have the clustering structure. But using our null data generation, the cluster is no longer there. And in this case, real data doesn't have clusters, and accordingly, our null data doesn't have clusters. So you could imagine, even if your procedure is clustering followed by DE, its results will be similar on null data and real data. So you wouldn't find DE genes. That's the idea. And indeed, it worked. So using this idea, we can see that our approach can control the actual FDR under the target across a series of target FDR thresholds. While using the naive approach, SURAT for clustering followed by any statistical test, the FDR is 1 because you have discoveries every time, and they shouldn't be there. So finally, I want to say that the take-home message is that sanity check is essential. We cannot just go with popular method. We need to think about setting up proper negative control, right? And also consider whether classical methods can do the job. And we have the simulator now, the third version, that can do multiple things. And double dipping, this is something that's ubiquitous in not just single cell, I think it's genomic data science, right? And finally, this is our proposal, and single cell simulator for generating the null data, negative control, and the Clipper that allows to control the FDR with just once null data generation. Finally, I want to thank my students like Vivian for starting the simulator work, and Tianyi for the version 2, and my current student Dongyuan for version 3, and also his other work I didn't talk about, and Guangao for generating reads, and also Xinzhuan and Kashim for this Clipper work and its application to the single cell case, and also the funding agencies. Thank you very much.
Great. So thank you for the quick talk, and I think we now head on to the discussion section.
Sure. I'll just sit here. Okay. Yeah. Yeah.
So I'll get started with the question, and then we'll take some questions from the audience.
Sure.
I think given that in these seminars we usually have broad audience,
Yeah.
Why should a data scientist be excited about the single cell technology?
Yeah, no problem. Yeah. Yeah, I think data scientists should be excited about single cell technology. It's because the data is very complex, and a lot of scientific questions cannot be answered just by eyeballing the data. You need some algorithm to help you analyze the data. And the increasing complexity will call for new algorithms. So I think there are a lot of opportunities there. And it can be from statistics to machine learning and even to just pure algorithm. Yeah, I think people can find the different angles to tackle the data analytical problem. So it's still a very exciting playground, right? People can find their niche and try to do something important. And also single cell data is probably the first time we can easily measure gene expression, genome-wide in individual cells, and also other features. So I feel like maybe new hypotheses can be generated, as we have seen some papers already, right? And how can we make the analysis better? So a lot of things to do. Yeah.
Any questions from the audience? Okay, then we'll keep the discussion going. So the take-home messages in your presentation are very useful for many data science domains. What would be a good checklist for somebody who wants to do inference on their data?
Inference on their data. Yeah. You know, traditionally, statistical inference, right, if we just use hypothesis testing as an example, it's about uncertainty quantification. So what can we say from your parameter estimates? Do we trust the nominal values, or do we want to assign some confidence intervals to it? You know, confidence interval and p-value are just two ways to assess the uncertainty. And I think here in this single cell data problem, it becomes harder because we have a lot of pre-processing steps before the statistical test, right? So then you cannot simply treat the problem as your textbook problem. We need some new methods, right? And so back to your question, what can we do? I feel like for the take-home message is that think about uncertainty, both bias and variance. Did your previous steps introduce bias? And did they increase the variance? I think these are always the things to think about. And I feel like setting up some proper negative control is a simple solution because you're not looking at one step at a time. You look at the cumulative effects at the end. Yeah. Yeah.
Okay,
we have a question. Yeah.
That was a great talk.
Thank you.
I have two questions. So in all these situations, you have to set up a negative control for
Yeah.
paper. What kinds of negative? So you were talking about how in single cell you can't just permute the labels because the clustering signature is still there. So for
Yeah.
other kinds of like genomic data, like microbiome or like metagenomics data, like what would be the negative control?
Yeah, that's a very good question. It's problem specific. I feel like the question would be different if you have your cells labeled using your knowledge. You know, okay, basically here the groups are predefined, right? Then you do the comparison. That becomes a standard testing problem. But here the case is that we don't know the groups beforehand. They were learned from the same data, right? So then you need to consider clustering as part of your procedure. So I think for other data, like microbiome data, I think it depends on what analysis is like. So that's why it's not a simple solution, right? It needs your talent to really design the inference problem. Yeah, and also inference is so difficult. Sometimes I feel like because it is about making a guess. And you could guess in one way, I could guess in another way. We just have to justify our guessing process. That's the statistical inference part. And why is it more trickier than machine learning prediction, especially supervised learning? It's because in supervised learning, you have some Y to help you do the error calculation, right? That's there. But here, inference is very subjective. Like frequentist inference, Bayesian inference can be different, right? They make their separate justification. So I feel like that's why the training is needed to really make the justification. It's subjective, and it's possible that no matter how hard we try to justify, we're still wrong. There's a chance we're still wrong. But at least I think having some justification is better than saying nothing, but just the results. Yeah.
Yeah, that's a very good point because
Yeah.
we were discussing earlier, like in biology, whenever we think about simulations,
Yeah.
we first think about, biology is so complex. How can we even, start doing simulations
Sure.
in
Yeah,
this space? But the idea is that we have to start somewhere.
Start somewhere.
Start with simple simulations, test our methods, and then slowly add
complexity. And also I think another use of our simulator is that because it's trained on real data, right? So it allows you to add perturbation to real data. And then you can still run the same pipeline and check the robustness, which is something I didn't talk about. Yeah. Thank you.
So problem of double dipping, actually.
Double dip, yeah.
It is a very prevalent problem in the genomic data science. It is also a prevalent problem in the machine learning
Yeah.
domain. So could you talk a little bit about why double dipping is a major concern and how to safeguard against it?
Yeah, it is a concern because you are using the same data to make multiple decisions. And the thing is that the secondary decision depends on the primary step, right? So in other words, let me just give a very good example, which is the motivating example for a topic called selective inference. If you are interested, you can check it. It's actually a solution offered by the statistics community, primarily I think statisticians at Stanford, to addressing a specific double dipping problem. So in their case, you can say, oh, let's say I have two sample t-tests, right? I first use a criterion to pick a feature with a small p-value. And then I want to say, oh, is this feature important? So then I just run one t-test for that feature. So you can see that I ignored that this feature was selected from many features in the previous step. It's possible that this feature is not that important because we know we have type 1 error. Then by random chance, you have many, many features. One of the features will fall into the tail for rejection. But then if you ignore that selection process, pretending you have only one feature, you will claim to be significant, right? So this is a very simple toy example to say why it is problematic. Because the data is random. But when you use the data once, the second time your decision depends on the same data, so you are using the noise basically from the data. Yeah, so in selective inference, they try to tackle this problem by modifying the testing procedure. Yeah, but I want to say that what's more challenging here is that the previous toy example is only about selecting features. But here the clustering is not on features. It's on your instances or your observations, whatever you call it, right? So it's on the random sample, not really on the features. That increases the complexity. And the paper, maybe I can go back a little bit since we're at the discussion. This one. So this paper from Daniel Witten's group, they were published in the top journal JASA in statistics. But I have to say, I don't agree with their formulation. The reason is that in their formulation, they consider it as a selective inference problem. They consider both the cells and the features be fixed. So what they consider as random sample is just the entries in the matrix, not the cells. Because when I read it, I was like, how could you assume the clustering to be fixed and be conditional? Because if I have a new set of cells, I do clustering. How can I say the new clusters are identical to the new old clusters? Because that's in their theory. Then I realized, oh, they fixed the cells. They are just assuming the entries to be random. But I think in the singles experiment, we shouldn't assume cells to be fixed because it's just a random sampling. You can measure these cells, not others, right? So I think that's the key thing. But such technicality can be not so apparent if you don't read clearly. Yeah. So here the challenge is that the previous steps operate on observations, instances, not just on features. So that's why the selective inference literature doesn't directly apply. Yeah. So I would say this is a very open problem, and we will need more people to realize the importance and get into the work. Yeah.
And another thing we were talking about is that in machine learning, you can always safeguard yourself from double dipping by having a held out
data
distribution test set.
Yeah.
But you don't have that.
We don't have that.
In this particular case where you're doing inference without that sort of...
Exactly. Because let's just imagine this double dipping clustering problem, right? What you could do is that you leave some cells untouched. You only use half of the cells to do clustering. And then you need to transfer the clustering rule to the new cells, right? To the left out cells. And you could do two sample testing on the held out data. But this doesn't address the double dipping because the rule is based on the gene expression. And then you apply it there. The bias goes with you there, right? So that doesn't solve the problem. And again, this because the operation is on cells, not on genes. Yeah.
So I guess we can go a little bit less technical now.
I
guess for the audience, I'm curious to know what prompted your journey in this direction of combining statistics with
and biology.
Yeah.
Single cell. Okay.
Yeah. Or biology.
Okay. I have to say I was a very curious child Yeah, when I was little. That's the whole start point. But I was like, when I was in high school, I knew I wanted to do something that can keep me learning continuously, right? And at that time, I couldn't think about too many jobs, honestly. And I feel like maybe professor is a good job for this. and then at that time, I need to choose the major to study in. And for my undergrad, I actually chose biology because at that time, it was just a time human genome sequencing completed, right? The very exciting time. And also, people say, there were a lot of open problems you could do. So I entered college, and I was fortunate because my college education was very broad. In the first 2 years, we took the same classes with other science majors. So that made me learn a lot of math and physics from the 2 years. And then at that time, my career has become the technology. And I knew that, probably quantitative biology will be the trend. So I knew that's the thing I want to do. And then I decided to learn more quantitative skills during my PhD. Yeah, so I feel like what I'm doing right now was what I wanted to do years back. Yeah. So that was fortunate. And also, I think the single cell problem, I have to say, maybe during my PhD, after PhD, I was looking for problems to work on. And gradually, I feel like my interests converged partly like this. I feel like, I want to understand more. Maybe after 10 years. After 10 years, I could step back and think about what is the problem or what questions I couldn't answer, not just writing papers, publishing. So I feel like, there are a lot of problems still very basic, not answered, even though we have 1,000 methods right out there. So, but still many basic questions were unanswered. Even normalization, we were talking this morning, right? Would you do log count plus 1 transformation, or would you do something fancier? What's the justification, right? A lot of basic questions were unanswered. And I believe many students will have the same questions when they just get into the field. So I feel like now my research is more curiosity driven. Yeah.
That's good to know that.
Yeah.
That's somewhere there, out there.
There you go. Yeah.
The opportunity to do that.
Yeah, definitely.
So then I guess building on this question, so for a data scientist, what do you think? So we talked about normalization, and so what are some of the other open challenges out there, and what kind of training could they do? Yeah,
that's a very good question, because I was at Berkeley for a Simon's workshop this summer, and I talked with Bing Yu, who is a very famous professor there, and she wrote a book called short as PCS. It's predictability, computability, stability. So she coined the 3 terms, and she thinks these are every data science project should focus on. So basically, the argument is that predictability makes you think, if a model can predict well, it is a reasonable model. And computability means that I could do the computation within a reasonable time frame, so it's workable. And stability means that the model doesn't easily collapse. So she said that these are the 3 principles, but how they should be executed in every project, she said that it's case by case. Yeah. So she said that scientists should collaborate with people with computational skills, computer science, statistics, to work together. And I was like, we couldn't expect scientists to learn so much, right? Because their expertise in the science. And she said that that's why we need communication, to use some common language to communicate. And I have to say, my personal experience is that I found collaboration with computational biologists is most effective, because they analyze data and they use algorithms, but their focus is on biology, right? So there's a common interest in the middle, and we develop methods, right? So if we collaborate, at least we can make sure, our methods have a user. And they will need what are the gaps, they will know what are the gaps in the current method. But if we skip computational biologists but directly collaborate with experimental biologists, I think that's more difficult. Because for them, some of the analysis, maybe even the very basic ones, right? Yeah, and they will, so the gap is there. But back to your question, what skills are needed? I think it's more and more difficult to know everything. The field is so big. So I think we should just be realistic and say we cannot know everything. But we should be specialized in the skills we're better at. I can say probably if you are more specialized in computer science, I think coding is very important, right? But in statistics, I mean, some type of philosophical thinking is important, like I said, frequent spacing, those kind of things. If you like it, maybe you can choose it. Or if you are very good at biology, if you're really interested in scientific questions, you can be more computational biology. I think choosing the speciality within this interdisciplinary research is very important. Then you find collaborators who can work with you. Yeah.
Speaking, when you said that we have to accept that we can't learn everything, that was one of the most humbling observation I made very early on in my career, that it's fine to not know everything.
Not know everything. Yeah.
And I think it's fine.
Yeah.
So, how important have collaborations been for you for your research?
I will say my recent work totally relies on collaboration, like the D-seq HR problem. If I didn't collaborate with a computational biologist, I wouldn't know it. Because the reason we figured this out is because these two tools are their to-go methods, right? So when they analyze data, they use them. Then we found this problem, right? And then because our paper has some quite high impact, let me scroll back here to that page. If you're interested, you could see we had some discussion. Okay, right here. This is my Twitter account. We have a very intense discussion on Twitter, actually, because some people were saying, oh, what we did doesn't make any sense, and we tried to explain and try to see whether it makes sense or not. But anyway, so I feel like many people, we have this intention on internet simply because so many people use those two methods. And when they saw, oh, these two methods don't always work, they became, wow, what do I need to know? Right. So I think that's a way to raise attention and also try to help people say, oh, at least the computational analysis is not just clicking buttons. We need to know more, right? So I feel like that's something we need. But even though I have to say, we were kind of brave to do this because nobody would like their methods to be criticized, right? But still, I feel like at least we're not saying the methods were wrong or doesn't work, but we're just saying they don't always work. You need to be careful.
Yeah. I mean, it's fine to understand the robustness of methods, and no method is perfect.
No method is perfect, and they were developed for small sample size, right? So that's why at that time, when you have such small sample sizes, you must make assumptions. Otherwise, there's no power. I totally agree. But now this analysis is in a different scenario. You have much larger sample sizes. Why would you stick with those methods, right? But if you don't have the training, you may go with popularity, and this is natural, right? Yeah. And I even received a comment on Twitter, which is very encouraging to me. He said, oh, I didn't know people care so much about method choice, but seeing your paper, I actually now see people do care about this. Yeah.
I mean, a big lesson from all of this that we've seen is that be very, very deliberate about the method you choose, not just because it's popular, but does it solve the problem the way you wanted to solve it?
Yeah. And another word I heard from my PhD professor, I think it's being used. She said that, oh, applied statisticians are not statistical analysts, right? So you cannot follow just a pipeline and do the data analysis. You need to put in a lot of your careful thoughts to justify the analysis. Yeah.
Absolutely. Yeah. Any questions or comments from
the audience? Yes.
So I was interested in what you were showing. I think it was like slide
22. Okay.
Like simulating batch effects.
Oh yeah. Yes.
Oh, no,
right here.
It wasn't
22 simulating batch effects. Okay, I think I know what you're saying. This one, right? Yeah.
So how do you get it to like basically like carry over from real data into like the simulated
data? It's just the parameters. So basically, I didn't talk about SDesign3 model because our time is limited, right? But basically, when we generate every gene's marginal distribution and joint distribution, we allow it to be conditional on the batch. So batch effect is treated as an intercept parameter in both the correlation matrix and also in the gene's marginal distribution. I think probably here we just use marginal distribution because we didn't feel the necessity to go to the next layer of complexity. But basically for every gene, let's say you have the gene follows a negative binomial distribution, conditional on covariates, and the covariates include batch. So it's like a generalized linear model setting. So the batch is part of the intercept parameter. So we feed that to the real data, and given the fitted model, we generate synthetic data. And over here, we set that batch intercept parameter to 0 to generate the right data. So there's no more batch effect. Yeah.
So could you see that being used as like a data augmentation?
Actually, yes. So when the model fits reasonably well to real data, you could use it as a batch correction method, right? So basically, once you set that parameter to 0, there's no more batch effects. And this idea is actually very, very similar in terms of, in the method you can check, it's called combat. It's a very popular batch effect removal method. Yeah. So they have combat and combat seek. So two versions with different models, but the idea is very similar. So they try to remove batch effects. But here, our major point is not that, but just the simulator. Yeah. Yeah.
Do you think though, could you, I don't know what I'm trying to say, like for like deep learning methods, like for example, if you're doing like a computer vision task,
Yeah.
it's pretty common to want to do some kind of augmentation,
Mm-hmm.
including some like random translations
Yeah.
to mirror it,
Yeah.
rotate it, something like that.
Yeah.
Do you think, could you use like this model in a certain way, in a similar way for
Mm-hmm.
single cell and AC data?
I know what you're saying. For data augmentation in computer vision, right, you take patches of the image, right, and you try to add some noise to it, and you use those together to have a larger training data set with more variations there, so your model training is more robust. I think that's the basic intuition there. But that is in a supervised learning setting. And so far, we haven't touched anything about supervised learning. But there are indeed some supervised learning problems in single cell field, like given the cells, given some cells with cell type labels, can you train a classifier so you can apply it to new cells to predict their types? There are maybe, I think, 20-something methods for doing that. It's called cell type annotation tools. And yeah, but people did question about their robustness because they may predict something totally unreasonable. So I think the recent development, I think there were some benchmark method papers. The recent development is that you should allow unknown cell types to be part of your labels. So then when you're not sure, you just predict unknown, right? And then I think your question will be, can we use our simulator to generate more, perturbed cells, add them to the training data to boost the robustness? I think that could be possible for that supervised learning task.
Mm-hmm.
Yeah.
Any other questions? Okay. So we usually like to end our on a happy thought.
Happy thought.
So based on for the trainees in the audience, based on your trajectory so far, what is your advice?
Wow. I was listening to a Nobel Prize winner's talk on Zoom like a week ago, and his name is Tom Cech. Tom Cech, he won the prize in 1986. Okay. 1986, yeah. 1989, no. And his trainee, he has a very famous trainee, Jennifer Doudna. Yeah. So anyway, so he got asked this question, and the audience, because it's a community talk, right? An audience, I think, is a high school girl asking him, "Oh, what is your advice? Because you're so successful, right?" His advice is like, "Oh, you should do the thing you are enthusiastic about, and don't listen to other people's advice." I think in terms of research, that's most important, right? Do the things that you are very enthusiastic about. But I have to say, honestly, I was so fortunate that when I joined UCLA, I really didn't try to adapt myself to the promotion criteria. I didn't really think, what I need to do to be promoted. Right. Because I felt like maybe I was confident, but I felt like if I couldn't succeed here, I would have good jobs waiting for me elsewhere. Yeah. So maybe not academia industry, but I don't think that's something I need to really force myself to become something unlike me. Right. But in the end, I was fortunate that I was in a place that was very tolerant and free. So yeah.
So be at a place that's tolerant and free.
Yeah, and also if you don't feel happy about a place, why stay there, right? You have a lot of opportunities here. Yeah, especially you are learning these quantitative skills that have a lot of applications, right?
Absolutely.
Yeah.
All right. Well, on that
Thank you.
note, let's thank the speaker again.
Thank you. Thank you. You're a fantastic host.
Oh, thank you. I appreciate that.
Yeah, fantastic. Yeah.
So, your next meeting with your
Soren in the